Alright, this video is going to be an introduction to all beta-lactam antibiotics and how they work. We're talking about penicillins, cephalosporins, carbapenems, all types of antibiotics, and they're all types of antibiotic families that work through a beta-lactam ring, which we'll get into here in a second. But the ones that you've most commonly heard of, like amoxicillin, ampicillin, ceftriaxone, cephalexin, things like that. All those drugs inside those penicillins family or that cephalosporins family, those all act through this similar mechanism. Okay, now we lump them all into beta-lactam rings or beta-lactam ring antibiotics because they all have in common this one feature, and that is a beta-lactam ring. So it's a four ringed structure here, four-sided ring here, with nitrogenous side to it here. The other key parts, double bond and oxygen inside that ring. Otherwise, this ring right here, this four-sided structure, is what all beta-lactam antibiotics have in common, right? So now, why is that important? Why do we call it beta-lactam? I mean, there's obviously lots of other parts to that structure, so why do we care about that? Bacteria have an enzyme called DD-transpeptidase. And, well, another big word, why do we care about that? When a bacterium, let's say this is our little bacterium here, when it goes to replicate, which bacteria do quite frequently, as you know, or even just to survive, it has this cell wall on the outside, right? Like, let's say this is actually our bacteria in here. And it has this cell wall, which like eukaryotic cells do not have, if you remember that from your microbiome class. But this cell wall needs to be broken down and remade, right? Because if it's going to butt off into two bacteria, or if it's just going to survive and just replicate and take out the old in with new, that sort of thing, parts of this get broken down all the time. It's constantly happening. And then they get replaced, right? I mean, like let's say we're making a daughter cell or whatever, it gets replaced. Well, how this replacement occurs, how these blue lines occur, is through this enzyme, DD-transpeptidase. It cross-links the building blocks of that wall so that you essentially fix those holes that have been made, right, as part of a natural process or just to survive. This transpeptidase, it cross-links the building blocks, and those building blocks are called, the other big word here, peptidoglycan, which, if you remember, is the main building block of the cell wall. So just to recap, bacteria have cell walls that are constantly being broken down and remade. Cell walls made of peptidoglycan. It gets cross-linked together by an enzyme called DD-transpeptidase. And how these antibiotics work is their beta-lactam ring, which is that four-sided ring that I drew earlier, inhibits this guy. So I'm going to draw a big old X here. And if we take out that enzyme, if it's not working, there's going to be no cross-linking occurring between the peptidoglycan. So there's just going to be holes inside this membrane. And if there's, holes inside this cell wall. If there's holes inside that cell wall, that cell's not going to be stable. It's going to be uncompensated, and eventually it's going to lyse, or it's going to explode or break apart. And thus that bacterium is no longer alive. So if we hit enough of these bacteria with beta-lactam antibiotics, they're going to lyse, they're not going to be able to replicate, that sort of thing, and they're going to die. And that's how all beta-lactam antibiotics, including penicillins like amoxicillin, ampicillin, penicillin, that's how they all work. Thank you.